Hi guys, this is Dr. Gonzalez here, and this is the first of several videos that you're going to have about inferential statistics and transitioning into t-test and then later ANOVA. I made the video last year and broke it into two parts, and I still feel like that was too much. So this year I've broken it down into four. So in this video, we're going to talk about what inferential statistics are. And then we're going to go into the basics of a t-test. Then in the second video, part 1 and part 2, we go over t-tests and how to calculate them by hand. And then in the final video, which is to me the most important one, I'm going to talk to you about how to run these particular statistics in Jamovi. When I get to that video, I'll remind you that there is a data set that you can download so that you can work through these tests as I go, because when you get to your assignment, that's going to be what you have to do. So let's get started. So what are inferential statistics? And I have the wrong notes, so I'm going to back out of those and go back to my other ones so that you have the right thing on the screen. So what it basically boils down to is there's a student version of the notes here, and I've taken out some of the bigger graphics so that you don't have to print out all of those big pictures that I just kind of have on the screen for extra, decorative stuff. So let's get into the final t-test notes, and this one should be Intro to Inferential Statistics. All right, so I started out by putting a cartoon. I took it out of yours, but in this particular cartoon, you can see that there are two people and they're looking at a scatter plot with a line through it. And they're commenting that they're freaking out because they found a correlation between owning cats and being struck by lightning. Now, as you know from PSYC 4370, the non-experimental class, we focused in that class on association claims. And association claims do wonders for setting us up for our experiments, because one of the prerequisites for an experiment is that you have already established causality. So not causality, I apologize, correlation. So you know that variable X and variable Y are correlated with each other. So in this particular cartoon, they're simply saying that owning a cat and being struck by lightning are correlated with each other. And as you can probably deduct from this, that is not a causal relationship. So we're going to talk about some inferential statistics within the boundaries of causal relationships. That's what we're trying to establish in all of our experimental designs we've been talking about. So in these videos, we're going to focus on how inferential statistics, and particularly t-tests, are going to be a tool for proving causality between two variables. So let's talk about inferential statistics. So what are they? Inferential statistics basically use data from a sample for you, the researcher, to make a decision about a population. So that's different from descriptive statistics, because in descriptive statistics, all we're doing is painting a picture of our sample. So we went over some descriptives in the last week. We talked about frequency distributions. We talked about measures of central tendency, mean, median, and mode. We talked about variability, which is standard deviation. Those all describe a sample, okay? And then we can hopefully take that sample and make some inferences about our population by using inferential statistics. The descriptors can't do that. Now, inferential statistics help us as researchers to estimate population parameters, to test certain hypotheses, and to assess relationships between variables. So by assessing relationship, we could look at a correlation, or we could do what we're doing in this course and look at causality. All while it's doing this, we're going to be able to account for uncertainty and variability. So our tests are going to be adjusted for those things. And I apologize, one of my cats is running through here like a crazy animal right now. So if you hear a wild cat in the background, it's Oreo on the rampage. So inferential statistics are going to be based on our sample data rather than population data. So remember, anytime we are sampling from a population, we want that sample to be representative because we want to have high external validity in our research. We want to be able to take the conclusions from that sample and put them back in the population. So that all hinges on whether or not our sample was actually representative of the population it was drawn from. Inferential statistics also involve probability theory, and we're going to talk about that in detail as we move through hypothesis testing. I know you're probably tired of hearing about hypothesis testing. We've talked about it within certain contexts. As we move to these specific statistical tests, you're going to see how our hypotheses change to reflect what we're doing in each test. So some of the things that inferential statistics would include would be the hypothesis testing, ANOVA, regression, correlation. Those are all examples of certain inferential statistics. What's the goal of inferential statistics? Well, there's several things you can accomplish as a researcher. First, we can use inferential statistics to estimate population parameters. So an example of that would be we could take a sample of college students and we could measure their height. And we could do this with 100 students. And hopefully that sample is representative of our population. So when we calculate an average height for that sample, we're actually calculating a population parameter using a subset that we pulled, that population. We can also use inferential statistics to test a hypothesis. So that hypothesis is a statement that's testifiable that is about the population that we're dealing with, and it's based on sample data. So an example of that would be that we could use inferential statistics to test whether a new sleeping medication is more effective than the standard treatment that's already on the market right now. Third, we can use inferential statistics to look at relationships or associations. So we could go back and we could use correlation as an inferential statistic to establish whether there is covariance between the number of hours of sleep and your academic performance. And then we can also use inferential statistics to make predictions about the future. And that could be future observations or future outcomes. So you have an example here on the video where you have a graph showing you per person spending on holiday gifts over time, and you see time is measured in years, so you're going from 2006 all the way to 2022, and you see the variation. And what we can see here is that there's an upward trend. Okay, you can see that in 2008 there was a significant drop, and that correlates to the recession that we had back in 2008. Let's pray we don't have another one of those again. But what would be the logical prediction if this graph continued to show data up until 2026? Well, based on the trend that we see, we would look for an anticipated increase overall as time goes on. Another thing we can do is we can take our findings and generalize them by extending that conclusion to a larger population. And inferential statistics will help us account for uncertainty in that. So an example of that would be using survey results from a smaller group to make a general opinion that's applicable to that broader population that we're pulling it from. So there are some points that you need to consider when it comes to inferential statistics. So first, you've got to remember that we create a question that's concerned with the entire population. But you have to continue to keep in mind that in a real research setting, you cannot sample everyone in your population. All right? The larger the sample gets, the closer it arrives to the actual population it was derived from. The converse of that is actually true as well. The smaller your sample is, the more it's going to vary probability-wise from the population it was drawn from. So the take-home message from that is that the quality of your sample matters. Another point to consider is that when we're dealing with inferential statistics, there are many times when we do not know the population value for our dependent variable. In a future video, I'm going to talk about a one sample t-test that is comparing a group of people's sleep duration on average in hours to a national average. And that's a special situation. Sleep duration is a construct that's been studied frequently. So we have a lot of sources out there that would let us draw a conclusion about what the average sleep per night might be. But there's a lot of times when we don't have that information. So you have to keep that in mind when you're making these broad claims about what you're finding in your study and how it applies to a general population. Now, larger samples are going to give you a more certain estimate. Every time you sample and increase the number in your sample, you approach the population a little bit more. Eventually, if you kept doing that, you would sample all of the population. That would become a census, and you would go to a point where it's not possible to do that. We cannot realistically sample all college students in the United States to determine how many hours they sleep per night. So what researchers do is, in order to get a better population estimate, they conduct multiple studies over and over and over again. And when you compile all those studies together, you're going to be able to draw a better population estimate. So those are just things that you need to keep in mind when we're discussing inferential statistics. Now let's go back and review null hypothesis significance testing, and we're going to refer to this as the NHST method. We're going to go through the steps of it, and we're going to kind of review how that plays a part in inferential statistics. So the null hypothesis significant testing method is a method that we use to decide if your sample's results were strong enough to say that there was a meaningful, statistically significant relationship about your population, or did what you find simply happen due to random chance. Remember, your null and your alternative hypothesis basically say the null, there is no association, that's a correlation. In inferential causal situations, we would say there is no effect of variable X on variable Y. Null, negative, nothing. No relationship, no causal effect. The alternative is a variation of the opposite of that. There is a correlation, there is a significant difference in a mean, there is a significant difference in a t-test, whatever. But it's the alternative of the null. And what we're going to do in null hypothesis significant testing is we're going to focus on that null hypothesis. We're not trying to prove that the alternative is true. What we're trying to do is disprove the null. So in order to do this, the NHST has a specific set of rules for you, the researcher, to follow when it comes to determining if your results were significant or not. Remember, your null hypothesis is going to always say that anything you observed that was a difference was due to chance alone. And that's going to stand to be true until it's been proven wrong. Okay? So here's an example of some null hypotheses. These are not necessarily related to psychology or human sciences. They're just a wide variety, so you can kind of think about hypotheses in a different context. In this first picture here, we have a picture of a cat, and the null says that cats show no preference for food based on shape. So I can show them fish-shaped food and triangle-shaped food, and they're going to eat it either way because cats want the food and don't care what it looks like. In the second situation, I have plant growth is not affected by light color. The supposition here is that it doesn't matter what color the lights are. If you put a plant underneath it, it's going to grow. And the third one here is that age has no effect on musical ability. What you will see in all three of these hypotheses, what they all have in common is the no, not, no. There is no effect. The plants are not affected. The cats show no preference. So the takeaway from that is that your null hypothesis is going to say that for whatever two variables you have, there is no significant relationship or no cause and effect relationship. And that's true for experiments because that's what we're talking about in this class, right? If I were talking about this within a correlational context, it would be a little bit different. So your research design is going to determine how you frame that null hypothesis. So remember, in an experiment, we're looking for cause and effect. So our null hypothesis is going to say that there is no difference in variable X in group A and group B. And that's assuming that X is your dependent variable, so it's the measured variable in response to your IV, and it also assumes that group A and B are going to be experimental and controls. The non-experiment would be that there's no cause and effect, but we are looking at an association or a correlation. So a null hypothesis in a non-experimental setting might say the null is there is no association between the IV and the DV. So here's an example. Maybe you have a biologist and that biologist is interested in testing a new biofertilizer. This is a fertilizer that is biologically based. So the null hypothesis would say there is no significant difference in the mean plant growth of the two groups. So I have one group that's getting the biofertilizer, one group that's getting no fertilizer or the regular fertilizer, and I'm testing some sort of growth, which would be, I guess, mean plant growth. We can measure that in height. So there's no difference. That means the means are not statistically different from one another. The means for those two different groups. Conversely, the alternative hypothesis is going to state that there was a significant difference. And what we're going to do is through the NSHT, we're going to set some cutoffs, some criteria for making that decision, and we're going to set those beforehand. So writing that alternative hypothesis would just say H sub A is there is a significant difference in the mean plant growth of the two groups. All of this should be familiar because we've gone over hypothesis testing already. Now, hypothesis testing in and of itself has 4 general steps. You'll find different versions of these steps. Some of them go into a little bit more detail. This is kind of the nuts and bolts of what has to happen in a hypothesis testing situation. First, we assume that there is no effect. So in the absence of data to prove opposite, we assume the null is true. Okay. Second, we're going to decide what statistical test to use, collect our data, and calculate our statistic. We also go back and calculate the probability of getting a significant and a non-significant result. And when we start using Jamovi, we kind of glance over or jump over this step. So that's why in this video and the calculation videos that follow, we kind of add this step back in so that you can see some of the things that are being done behind the scenes in Jamovi. And then finally, what you're going to do is make a decision. You're going to decide whether to reject the null or fail to reject the null. Sometimes you will hear people say retain the null for the fail to reject. I want you to try to move away from that way of talking when it comes to the null hypothesis. You can reject it or you can fail to reject it. Rejecting it means that your alternative has been shown to be more adequate, and failing to reject it means there was no difference and you're going to keep it. It was true. So let's work through an example of a study that is examining the reduction of screen time and sleep quality. So the first thing you're going to do is you're going to assume that there is no effect from your treatment. We're going to start off by being pessimistic and assume that our intervention doesn't work, and that is the nuts and bolts of your null hypothesis. Think of it as the glass is half empty. So using our example research question I introduced on the previous slide, our null hypothesis would be H sub 0 is there is no effect in sleep quality by reducing screen time before bed. So variable X, reducing screen time, does not have any effect on variable Y. Variable Y is sleep quality. On the other hand, your alternative hypothesis is going to state that sleep quality was improved by reducing screen time before bed. Our manipulated variable is screen time, and your outcome or measured variable is going to be sleep quality, and it's going to be improved. So technically, this is going to be a one-tailed hypothesis. Going into the next portion of the procedure for hypothesis testing, we would decide what statistical test we're going to use. You'd collect your data, and then you would calculate your statistic. So what test you use is going to be directly the sum of what research design you had, your operational definitions, your measurement tools, and how your statistical design plan all work together. Those four things inform which test you use and how you calculate that statistic. Now, in the example that I gave you, you were looking at mean difference in sleep duration in two groups. One group got the intervention, and the intervention in this case was no screen time. Then you had a group that did not have an intervention. Sometimes it might be a control group, sometimes it might be phrased as the placebo group. But in this particular research question, the control group was allowed to use screens with no limits. Continuing in the process, you would then calculate the probability of getting a significant and a non-significant result. And that sounds very complicated, but it's going to be really easy when we break it down. What you're basically doing here is you're looking at this distribution, okay? And you're deciding where in the distribution your score has to fall in order for you to reject the null or fail to reject the null. And you're setting some specific criteria for that. So this little area under the curve is where your test statistic would fall if you reject the null. And the corresponding p-value to that statistic is going to be less than 0.05. 0.05 is normally what we choose. There are other levels you could choose for this class. We're going to stick with 0.05 because it's easier for everybody to remember it if we're on the same page. So again, we're going to calculate that probability. If it falls within that critical region under the curve, this pink area in this particular distribution, we reject the null. And then if it falls in the rest of this, we would fail to reject the null. I actually need to go back and change that language. I used retain the null because it was the language we used 20 years ago when I was taught this, and I'm sorry, it sticks in your mind. Since then, we've gotten away from saying retain the null, and we prefer the term fail to reject. It's just very hard for me to get that to stick in my mind. After you've done that, and you've looked to see where your particular statistic falls on the distribution, you can decide whether to retain or reject. You're going to actually make this statement in your writing. You're going to reject the null and say there was a significant difference. You're going to report your statistics. We're going to talk about how you would report those statistics in the future videos. Then you would also say fail to reject, and I said some people say retain. And that is when we have no significant difference between the two groups. Let's talk a little bit more in depth about statistical testing here. So there are a ton of different statistical tests that you can do. There are entire courses that are based on what kind of test is needed for an experiment, for a non-experimental study, for a qualitative study, for a quantitative study that's an experiment, a qualitative study that's non-experiment. It goes on and on and on. So how do you know what statistic to use? Well, this goes back to your research methodology. We talked about within and between subjects. When we get to the different versions of the t-test in a little while in another video, we're going to associate those research designs to the appropriate statistical methods so that you know what's expected of you when you're writing a proposal like you are in this class. Some experimental methods might be t-tests, ANOVA, regression, and mixed models. Now, if you've been in PSYC 4370, you'll recognize that regression is also a non-experimental method. So it's all dependent on the situation. Two other non-experimental methods we cover in 4370 are correlation and chi-square. All right, so to determine if your results are significant and reliably different for other results, you're going to define your null and your alternative hypothesis. You guys should be able to do that in generic words. These next videos are going to help you add the statistical language to it. You also should be able to determine what statistical test that you need to use. You are going to learn how to calculate your statistic, and you're going to also state your significance or alpha level, and then you're going to go into the rest of the procedure by calculating your degrees of freedom. That's going to help you establish your criteria for rejecting and failing to reject the null. You will state your decision rules, and you may have to use a table if applicable. You might use in Jamovi just which p-value would be significant, which would not. Then you're going to state your results, and then in a paper, you're going to draw a conclusion and discuss based on those results that you got. So if we go back to our example research question, we talked about, does reduced screen time increase the duration of sleep in young adults? So I just have a little graph showing you here what we're looking at. You've got sleep duration on your y-axis. You've got the intervention on the x. Your intervention is no cell phones versus cell phones or electronic device screen time. And what you're seeing here is that sleep duration, assuming this is hours, was higher for the group with no screen time when compared to the group that did not get the intervention. So again, we're answering the question, does reduced screen time increase the duration of sleep in young adults? You've got group 1, no electronics. Group 2 has electronics. Your null and your alternative hypotheses, the mean of group 1 is equal to the mean of group 2. Now, this is going to be true in a specific situation for certain t-tests. When we get to each particular type of t-test, I'm going to walk you through how your null and alternative change. This is just a generic presentation of what happens in this particular research situation. Your alternative would be that the mean of group 1 does not equal the mean of group 2. Now, this particular hypothesis is not indicating a direction, so it would be two-tailed. Next, you would determine your test statistic. If we had 20 participants and they were randomly assigned to those two different groups, we would go ahead and assign them. They would do the intervention, and then afterwards we would measure their sleep duration, and we're going to measure that in hours. So what this is actually, as far as design is concerned, this is a single factor design with two levels. So your single factor is sleep duration, and it has two levels. You can either use electronics or you can not use electronics. So the appropriate test to analyze a single factor two-level design would be a t-test. You've got two groups, and we're looking at one particular factor broken into two levels. So t-test is appropriate for that. In the third video, or the fourth video actually, we will get into when to use a one sample t-test, when to use a two sample t-test, and when to use a repeated measures design and t-test to achieve the results you want in your research question. So that is the end of the first video. The notes for this will be published in Canvas as well. Once you're finished watching this video and kind of digesting this information, you can move on to the second one.